Hello everyone, morning. I'm Weiyi Tang. Thanks Brian for the introduction, and thanks for joining for organizing the seminar, even though he's not here today. And it's my great pleasure to kick off the faculty seminar. I'm going to talk about some of my recent work regarding how the substrates regulate contribution of ammonia and urea to marine nitrification and nitrous oxide production. So I would first thank my collaborators from Princeton University, where I did my postdoc, and also colleagues from Xiamen University, and also appreciate the funding support from NSF and Simon's Foundation. Since I'm pretty new over here, I will give a very basic background about what I have been working on and what we're doing here at CMS. So I'm generally interested in studying ocean changes, and particularly under both natural need and under project activity in the future climates. So the ocean is getting warmer, it's becoming more acidic, and it's losing its oxygen. So among all of those changes, I'm particularly interested in the biogeochemical cycles, including the carbon-nitrogen cycle and carbon cycle, in which they interact with each other. So those biogeochemical cycling processes are significantly perturbed by anthropogenic activity like fossil fuel combustion or application of fertilizers. And those cycling processes also provide feedback to the Earth's climate system, such as through the regulation of greenhouse gas emission and uptake. And I use a variety of approaches to study those processes, including both geochemical isotope measurements and molecular biology techniques like omics that I will talk about today. And especially I will be setting up a lab that can measure isotope trace amount level for both nitrogen and carbon. I'm a seagoing oceanographer. I collect most of my sample from research cruises. I also use remote sense techniques like biogeochemical algal floats and remote sensing to look at ocean changes at larger scale and temporal scale. But for today, I will be focusing on the nitrogen cycle. So as we all know, nitrogen is essential for supporting life on Earth. And nitrogen is a building block for many key biomolecules like DNA, amino acid, and protein. At larger scale, nitrogen really regulates the Earth's ecosystem productivity, and that in that process supports the food production and CO2 uptake, for instance. As you can see, as bottom panel, that the surface chlorophyll distribution really follows the surface nitrate distribution, right? So considering the ocean changes, it's really important to understand how the nutrient distribution and how the nitrogen availability will change under future climates. However, in addition to nitrates, there are so many other forms of nitrogen present in the ocean, and this is only a simplified view of the nitrogen species. In addition, there are diverse group of microbes present in the ocean, transfer different nitrogen species and regulate the amount of nitrogen available to phytoplankton or other microbes. For example, nitrogen fixation converts nitrogen gas into ammonia and then into biomass. This is the largest external nitrogen source into the ocean. And ammonia and nitrates can be assimilated into phytoplankton biomass. This is a process called primary production. And some of those organic matter will be remineralized back to ammonia. And other fraction can be exported into the deep ocean called biological plant. Right? During this transport, the organic carbon or nitrogen will support the deep biome, and those organic nitrogen will be remineralized back to ammonium, and some of the ammonia will be oxidized to nitrites and then to nitrates in a process called nitrification. So that's one reason the deep ocean is dominated by nitrates. This process also produces a very strong greenhouse gas called nitrous oxide. Some of the nitrate will be mixed back to the surface and support the phytoplankton. However, under low oxygen conditions, such as oxygen minimum zones, denitrification and NMOX will convert those ammonia or nitrate back to nitrogen gas, meaning reducing the amount of bioavailable nitrogen in the ocean. So you can see this, even though this very simplified view is pretty complex, but what makes the nitrogen cycle really fascinating is how it interacts with other elements, such as carbon, phosphorus, iron, and etc. However, the current nitrogen cycle are severely perturbed by anthropogenic activities. For example, the first-of-field combustion emitting nitrogen-containing particles into the atmosphere, and that can transport and being deposited into the ocean. The excess nutrients into the coastal water leads to eutrophication. The ocean is getting warmer, and intensified eutrophication might inhibit the supply of nitrogen from the deep ocean to the surface. The expansion of the oxygen minimum zone will potentially enhance nitrogen removal. In that process, reducing the amount of nitrogen available to productivity. So I have been working on all aspects of the nitrogen cycle, and I will be working on how to quantify the response of nitrogen cycle to different environmental forcing. But for today, I'm only going to focusing on a process called nitrification in the red circle over there. And more specifically, I'll be talking about the first step of nitrification called ammonia oxidation to nitrites. So this is a process transform nitrogen different species, and it is autotrophic, meaning it takes CO2 into organic carbon. This process, as I mentioned before, is a major source of N2O production. N2O is a powerful greenhouse gas, around 300 times more powerful in terms of radioactive forcing compared to CO2. It also destroys atmospheric ozone. This process is conducted by microbes called ammonia oxidizers. Those microbes, the ammonia oxidizers, are among the most abundant microbes in the ocean. Although this process is called ammonia oxidation, however, recent studies have found ammonia oxidizers can use organic nitrogen, and particularly the urea, as a substrate to perform ammonia oxidation. In here, this is a study by Rafter et al. Conducted a time course study in the California system. So what does it found over the 2 years? Sorry, it's not working. So you can see the red line is the urea oxidation rates, compared to the black line is ammonia oxidation rates. The magnitude is quite comparable, right? Meaning if you're only looking at the ammonia oxidation, you will miss a lot of the nitrite source from urea, right? But the question remains whether N2O is being produced from urea oxidation, and what's controls the distribution of urea oxidation, and how important is urea oxidation globally? Because this is just a study from California system, right? So to answer the first question, I performed field work and to measure N2O production from urea oxidation, and for the address this second and third question, I performed the meta-analysis of all the currently available data sets to address how important is urea oxidation globally. So let's first talk about the field work that I conducted in the Chesapeake Bay. So Chesapeake here is CMS. So Chesapeake Bay is the largest estuary in the United States and located in the East Coast, surrounded by DC area and Maryland, Virginia. So one of the reasons I picked the Chesapeake Bay is because not only because it's huge, but also because it represents many other estuary and coastal environments in the world, because it covers a wide range of temperature, nutrients, and oxygen conditions. As you can see, in the summer, the bottom water of Chesapeake Bay experiences hypoxia or even aloxia, such as in this mid-deep channel over here. So in August 2021, I went on a cruise to collect samples from upstream to downstream at those 4 different stations to measure nitrification, urea oxidation, and N2O production. I wasn't going to use CB2, these sites, as an example, but I did measurements across all the sites. This is the distribution of ammonia and urea concentration. Ammonium generally is very low at the surface and increase towards the bottom water. It's not that deep, it's like 20-25 meters. And urea concentration is around 0.5 to 1 across the water column. If you look at the N2O, the gas, greenhouse gas, It's generally around 9 nanomolar concentration, and then it decreases towards the bottom toxic or hypoxic water. However, this red dashed line is the equilibrium concentration of N2O with atmosphere. So you can see almost the surface N2O is oversaturated, meaning there's less production of N2O, and that can potentially emit into the atmosphere, right? So that indicates this N2O might be produced from nitrification. So I picked 4 different depths to conduct incubation experiments. Specifically, I was using N15 labeled ammonium and urea injected into the incubation bottle. After around 12 hours, I tracked how much of those tracer being converted into N15 labeled nitrites and N15 labeled N2O, right? Just step back a little bit, and in the natural form, most of nitrogen is in the form of N14, right? So if you add those N15 tracer, it can really help to increase detection or increase the sensitivity of your measurements. So here I'm going to show you the distribution of those ammonia oxidation and urea oxidation. On the left side, it's the nitrite production from ammonia. This is nitrite production from urea, right? So you can see they are quite distinct vertical distribution. The nitrite production from ammonia generally increase with depths. For urea, a decrease, but there's a subsurface peak, right? But most striking difference is actually the magnitude. If you look at the magnitude, here the maximum is around 900. This is around maximum is 12, right? Considering the concentration is not that different, right? It's maximum ammonia is 3, the of urea is around 1, right? 0.5. Right? And similarly for N2O production from those 2 different substrates, you can see the vertical distribution is different, and the magnitudes also varies a lot. And N2O production from urea shown in the red orange line, 1 to 2 magnitudes smaller. Right? So I performed those same experiments across those 4 different sites. I'm going to summarize it. I'm not going to show you the depth profile, but I'm going to show you the contribution, right? So here I'm showing this is 4 different sites, and those are the depths. At some depth station, I have 3 depths. At this station, I have 4 depths. Okay, so those are stacked plots and color coded by the 2 substrates: ammonia versus urea. Ammonia in blue, urea in red. Just the same color scheme, and overall is 100%. Right. So this is the contribution to nitrogen substrates. Okay, and what you can see is generally the bluish color, the contribution or the fraction increase with depth, right? Generally speaking, similar to CB2, okay? And overall, urea accounts for 7 to 92% in terms of concentration. However, if you look at its contribution to nitrite production, right? That's ammonia oxidation versus urea oxidation, okay? It only accounts to 0 to 24%. And if you look at the N2O production, very similar contribution to nitrite production, 0 to 24, right? And meaning urea oxidation is occurring in the Chesapeake Bay, but its contribution to nitrite N2O is very small, relatively speaking. But you do see in those locations where urea contributes more, there tends to be more contribution to nitrite N2O as well. So what I did is I assessed what's the factor driving its distribution or contribution, right? So one of the key factors that we found actually is the urea to ammonia substrate ratio. This is the ratio in the log scale. On the y-axis is the urea oxidation divided by ammonia oxidation. Again, this is the ratio. Okay, I'm going to show you those kind of ratio plots a lot in the following slides. Just keep this in mind. What you can see generally is a positive relationship, right? Even though it's a log scale. But the other thing I want to point out is you can see the urea can be higher than ammonia, right? Up to like 10-fold. But most of the rates are below 1. The ratio of the rates below 1, meaning urea oxidation is smaller than ammonia oxidation. Right? And similarly for N2O production from urea versus from ammonia. Again, there tends to be a positive relationship. And NTO production from urea is relatively small. Okay. So if you look at this, at least I've shown in the Chesapeake Bay, urea can be a substrate of nitrites, can contribute to NTO production. And their contribution is proportional to the substrate concentration. But how does it work in other regions? It is just Chesapeake Bay or the same pattern in the globe ocean. So here, what I did is I compiled all the measurements of ammonia oxidation and urea oxidation either by my colleagues or from other groups. Here is the Chesapeake Bay in the cyan color. And there are some measurements in the Georgia coast, northern Gulf of Mexico. This is California, the study I mentioned before, Arctic, Antarctic, and many measurements in the Western Pacific. And now I'm going to show you the ratio plots. And keeping this in mind, okay? Y-axis, again, urea oxidation divided by ammonia oxidation. X-axis is the concentration ratio. So overall, you do see, again, there's a positive relationship, okay? And another important thing is majority of the data points, right, is below this one ratio line, right? Meaning urea oxidation is generally smaller than ammonia oxidation across the globe, except in the region is very high urea to ammonium concentration ratio, around 10, for instance. And if you dig into details, Chesapeake Bay is right over there, a lower urea to ammonia ratio. And this is around California system. And here is the North Pacific. You see this propagation, right, from more like eutrophic system, estuary to coastal and to oligotrophic ocean. But there's some weird numbers over there, and that's the Arctic and Antarctic. So what really stands out, despite this lower substrate ratio, right, compared to the Chesapeake Bay, and they have high urea oxidation rates in comparison to ammonium. This is right, intriguing in all the ways, right? Why there is supposed to be a positive relationship at the beginning, and why does polar ocean stand out, right? So let's talk about the ammonia oxidizer a little bit, right? Imagine, does it mean this ammonia oxidizer in those environments prefer urea over ammonium in the polar regions? Let's find out. And so I will give a little more background information about those ammonia oxidizers. Okay, so we use AMOA gene. That's the gene encodes enzyme to prefer ammonia oxidation, and it can be used as a marker gene for the abundance of ammonia oxidizer, or quantify or characterize what's the microbial community of those ammonia oxidizers. And similarly, the URAC gene is a marker gene for urea hydrolysis, right? Meaning if you have more URAC, and you can potentially utilize urea more efficiently. If we do a ratio thing, again, the ratio, URAC divided by AMA, it can potentially use an indicator for the ammonia oxidizer's ability to utilize urea, right? If you're ammonia oxidizer, meaning you have AMA gene. If you have more URAC gene within yourself, you potentially have higher ability to utilize urea, right? So now I'm just giving you a short, quick quiz, right? Imagine, and you across different environments, you have variety range of URAC to AMA gene ratio. Okay, what would be we expect how the urea oxidation divided by ammonia oxidation ratio would be? Well, it would increase or decrease, considering URAC is an indicator, or the ratio of the gene is an indicator of capability to use urea. Just give, oh, my own.
I'm like, how do you describe that? Like that?
Oh,
Like
yes.
Linear positive relationship.
That's exactly what I would
Not
expect.
caffeine yet.
Yes, thank you. So, but let's find out whether this is true or not, right? So the approach I applied is metagenomics. To look at some of metagenomic sample collected in this ocean to determine this URAC AMA gene ratio. So metagenomics is an approach that can reconstruct microbial community and the metabolic function of organisms in certain samples. For example, in the seawater, from the soil, even from human guts. It's widely applied now in medical science, for instance. So I'm going to briefly go over how it works. Imagine you have a mixed microbial community in this like seawater samples, and we collect into filters, and then I extract the DNA, and those colored circle represents different species. That's the genome of those different species. And then in order to get the genetic information, the sequencing facility sequence there and get the short rates, right? Usually the 150 or 250 base pairs. However, we cannot get much information from short rates, right? So there's a lot of bioinformatics tools to put into longer rates or longer sequences, and eventually trying to form those metagenome, assembly genome, right? So this process is really like to solve a jigsaw puzzle. You start with small pieces, and you can put everything together and form a whole picture, right? So the advantage of this is really figure out, well, if the ammonium oxidizer is present, and if these ammonium oxidizer contain uracil, right? So I applied this approach to some of our new samples in the Chesapeake Bay and in the Western Pacific. And also I put together some of those medical genome samples from previous study in the region where those rate measurements are performed. Although they are not always overlapping, but I try to cover the representative region, for example, in the Pacific, Atlantic, and Polar Ocean. So that's the distribution of this uracil AMA gene ratio color coded. Okay. If we first look at the Chesapeake Bay, has cyan color or bluish color, meaning the ratio is very low. Basically, uracil is not present in ammonium oxidizer. Similarly, in the Georgia coast, northern Gulf of Mexico, if we move towards offshore a little bit, and in this oxide minimum zone, you can slightly see more reddish color or bright bluish color. Particularly in the South Pacific and Western Pacific, the ratio can be 1 or even higher, right? So it looks like there's a spatial variability. And what we found, what driving this spatial variability, this substrate ratio is really good predictor. And so y-axis is the gene ratio, x-axis is substrate ratio. I work on to project all the data point here. This is Chesapeake Bay. Here is Georgia coast. This is Gulf of Mexico. This is Eastern Tropical North Pacific and South Pacific oxygen minimum zone. Over there is North Pacific, Pacific and Atlantic. Overall, you do see a positive relationship over there, right? And what's really interesting is, again, Polar Ocean stands out again, despite the lower urea to ammonia ratio, they have elevated the capability to utilize urea in terms of the uracil to AMA gene, right? Does it ring a bell or remind you something? Right? If you look at the rate ratio, that's substrates, that's the rate ratio. The Polar Ocean also stands out over there, right? That's kind of fascinating, right? And what I want to put this together is, if you look at left side figure, right, that I showed before, gene level substrates is more like microbiology. Right? If you look at, if you look at the right hand side, that's the rate measurements and substrates. That's more like geochemistry. If you put those two together, I will call biogeochemistry. So the y-axis is the rate ratio, x-axis is the gene ratio, uracil divided by AMA, right? If you look at put all data, there tends to be a positive relationship, right? So meaning we can potentially predict or give some indication what the microbes are really doing in the ocean based on their genetic capability, right? So that's our prediction on Maya's prediction, and looks like most cases is and matching. However, there's still a lot of gaps, right? So I'm just going to quickly summarize my talk, and we found urea ammonia substrate ratio is a good predictor of ammonium oxidizer's capability to utilize urea. And we developed a predictive framework between the substrates, nitrification rates, and N2O production, which hasn't been recognized before. But I still want to emphasize that there's still limited observation of the particular for N2O production. There's only one observation from Chesapeake rates and we did. And so Bob and I actually trying to develop instruments to measure ammonium and urea concentration at high resolution. And then we can couple to the stable isotope approach, trying to test this predictive framework more broadly in the ocean. But how does this everything ties back to my overview of the nitrogen cycle and perturbation? I have shown you urea oxidation can be important to nitrification and it being regulated by the substrates, right? But considering the fertilizer application, there's more urea fertilizer being applied compared to ammonia or nitrates, which can potentially change the ammonia urea substrates concentration in coastal water and potentially shift the ammonium oxidizer community towards using urea. Right? And with that, I will just put this summary slide here regarding what my lab does, and I'm happy to take any questions.
Presenting a lot of work that we've been doing monitoring sargassum in the Central Atlantic and in coastal. Oh, sorry, I gotta start my own timer.
Nice boy.
Of course, lots of other people contributed to this work and actually did most of it, and I'm just somebody who gets to talk about it. And then also funding from EPA and NASA and NOAA have all contributed to the progress that I'm hoping to show. All right. First, the background. We are the Optical Oceanography Lab. This is a very recent picture. There are 7 students-ish, 5 postdocs, 2 staff, and then myself and Chuamin are the faculty representatives of this group. So we do optical oceanography, which in real basic terms is how does light interact with the water, stuff in it, and stuff on it. We do this from in situ platforms. We go out and take measurements of light interacting with water. And we also primarily, what I'm going to be talking about today is we use satellite data sets to do essentially the same thing. Okay, so Sargassum. If I was giving this presentation 10 years ago, which I did not do my dissertation on Sargassum, but this is what the picture for Sargassum looks like 10 years ago. You have, it is the Sargasso Sea is essentially the North Central Atlantic. It is named for the macroalgae that is the primary environment inside the Sargasso Sea. And in the Sargasso Sea, you can find clumps of Sargassum. We have 2 different species in the Sargasso Sea. Those are they're small morphological differences between them, but overall they both float at the surface and they have a long history in the region. We know this not just because some of the earliest explorers traversing the ocean recorded it, but there are also communities that have evolved mimicry and so forth, evolved to live within and among the Sargassum. This includes fish, turtles, invertebrates, reptiles, you name it. There's a lot of community within this environment. Over the last 10 years, the picture has changed substantially. Now we find not just Sargassum in the Sargasso Sea, but also a huge Sargassum belt that is in the kind of the North Tropical Atlantic. It ranges all the way from Africa to through the Caribbean into the Gulf of Mexico, and you see it going out to the Gulf Stream. Usually the way that you hear about Sargassum now is in pictures like this here, where you have huge aggregations that are amassing on inland or on islands and coastal regions throughout the Caribbean, and it really wreaks havoc on these environments. Okay, so with that history, the long history of the co-evolving with the resident flora and fauna, but also with that kind of devastation that you see in the coastal environment, I'm often asked the question, is Sargassum good or bad? And I mean, clearly the answer is yes, but to add a little bit more meat to that bone, you know, we talked about the offshore and the pelagic realm. It's very helpful. It's got all these communities that live in it, and it's actually considered essential fish habitat, which means you can't just go out and take it because there are so many species that rely on it, including 3 Endangered Species Act turtles. Sea turtles. As you get into the nearshore realm is when you start to see problems. And the very first thing that you can have, even without physical interaction with the nearshore environment, is just shading. There are a lot of things in the coastal environment that need light to come down to that. Seagrasses, sponges, corals, algae, and so forth. They can live on the bottom and be shaded out by sargassum just by it being in the surface water. And the point I want to make with this picture here is you see these, you can't see the bottoms of these people that are swimming in the water. And if you look at the bright sand here, you know, the shaded region is exceptionally dark. So this can really, if it's prolonged, it can cause some pretty severe consequences just being in the surface water alone. Once you get farther up onto the shore, though, you start to have problems amplifying and multiplying. So you often see beach environments that have piles and piles of seaweed that have amassed. Any of those organisms that were living in the seaweed, the sargassum, they may not be able to get back out into the water, and they can die and sink. Sea turtles that use beaches for nesting, they can get this can be an obstruction for them trying to get out, lay their eggs, and come back. And juvenile sea turtles inside the sargassum. Also, this can be a problem. And I want to highlight that picture on the right there is a mangrove island. You see sargassum, we often see just the beach pictures because beaches are really front of mind for many people. But imagine getting sargassum out of those mangroves, out of the interstitial space between the mangrove roots. It's actually quite difficult. So just because we often see it on beaches doesn't mean that's the only area that's problematic. Another follow-on effect, once you have something stagnant in the water or right on the coast, it starts to decay. And just like with any, brewing tea, you start to get the decay. It starts to release nutrients and DOM and so forth into the water. This eventually creates typical eutrophication. You have eventual hypoxia, anoxia, and so forth. And you can actually visualize this with these patches that are near shore here, and you can see the plumes of discolored water kind of emanating from this rotting seaweed. This entire area could be that turquoise color, but it's not. Oh, sorry, I also forgot to mention it releases a hydrogen sulfide. Let me go back to it, actually. It releases hydrogen sulfide into the air, which stinks very, very bad. It can cause respiratory distress in some, and it really impacts tourism for a lot of these communities that rely on the ecotourism. And there is arsenic in the tissues. So that complicates some of the cleanup and remediation efforts. What do you do with thousands of tons of rotting macroalgae that might be toxic? And this is kind of what that looks like, at least on the front end. You have multiple people that can manually pick up, shovel this stuff, put it into front end loaders or dump trucks or something. They generally take it away and just dump it in a landfill because that's what we do. But often, or it's important to note that you don't just pick up seaweed itself. You get sand and other things with that. So this also problem contributes to beach erosion and so forth. If you are wealthy enough to be able to install something like this, this is a boom that can trap the sargassum offshore, prevent it kind of from coming inshore, and then you can collect it again before it comes into the nearshore realm, ideally. Another option is like you see in the bottom right there, where you kind of set up a barrier and kind of shunt it inshore, and then you collect it from a shore-based location. It saves you from having to get it from the offshore environment. Still very expensive, very difficult to do. And even if you do that, you still, notice there, that's that sargassum brown tide. So you still have some of the follow-on effects, even if you're able to remove the sargassum itself. Okay, so I said we do we do optical oceanography. So that's how does light interact with things on and in the water. And what I'm showing here is essentially the type of measurements that we take to measure something like sargassum. So on the left, you have kind of microcosms that we set up that have different densities of sargassum coverage. And then on the right are the light that's reflected off of that surface. And the main point I want to make is as you get denser and denser sargassum, you get kind of this elevated going up reflectance. And particularly in a region that is right here, this one right here, where all these arrows are. That's the red edge, what we call red edge. Almost all vegetative matter has a elevated reflectance kind of right there in that deep red, early near infrared area. So that's the main signature or feature that we use, not just to identify that sargassum is there, whether or not it has that little shoulder, or not just that it's there, but as well as how much of it is there. Look, if you notice that red edge feature increases as the density of the sargassum increases. Now I'm going to use a jargon term here. It's called AFA. That's the way that we measure, the way that we quantify that red edge peak. You don't need to know what it stands for. I just want you to know that that's the measure that we use to capture that sargassum abundance. And this is what we do. So we have satellites that are looking down. They're looking down on Earth, crossing over every multiple times a day. And they allow us to look down at the oceans, see if we can identify this red edge, this A-phase spectra, and identify that such that we can say relatively definitively whether something is sargassum or not. So a positive detection would look like this. So in the brighter colors are sargassum there, and the spectra that is associated with that, as long as what the AFA, that's there's elevated AFA for the sargassum features you see in that middle panel. But of course, it's never that simple. There are other things that make an elevated AFA signal. One of them that will be very important for this talk is just nearshore water. When you're close to land, you get elevated AFA pretty much universally. Another feature that you see a lot in satellite imagery that also has elevated AFA is just cloud shadows. So what our lab does is try to develop algorithms that can both use the spectral data, so the characteristics of the light coming off of the features, as well as the spatial characteristics. So we know sargassum arranges in these kind of like streaks in the images. So we develop machine learning algorithms that will identify the spectral and spatial features and identify them as sargassum or not sargassum. And from that, we can build things like this, this is looking at the entirety of the Great Atlantic Sargassum Belt. This is the 2024 version. Usually it starts actually in late the year before, so it starts in October of 23. But you can see the abundance of sargassum throughout the entirety of the world's oceans as the Atlantic Ocean through time. Brighter colors mean higher abundance. And generally what you can see is at the start of the bloom, you have a relatively small, but maybe 1 million metric tons of sargassum kind of off the west coast of Africa. And it builds and grows into the growing season, just like seasonality, just like most plants. It migrates westward with the prevailing currents. And once you get into kind of the March, April timeframe is when you start seeing real impacts into the Caribbean Sea and eventually into the Gulf of Mexico and Florida Keys and so forth. In 2024, we didn't really have anything in the Florida Keys. Not a bad condition, but that's why you don't see it migrating all the way up there like you saw in some of the other animations. So this type of picture is actually the platform for what we call our Sargassum watch system. And through this, every single day, I mentioned there are several satellites that are passing overhead and collecting data. Well, we grab those data, we use our machine learning algorithms to identify sargassum, and we provide those information online to various stakeholders. And so each of the white boxes in this representation here is an individual station. So I could click on the Eastern Caribbean region there, and it would take you to all of the underlying satellite data that were used to comprise that composite there. We don't just do sargassum, of course. So we have other products that you can see from chlorophyll, or from satellites: chlorophyll, sea surface temperature, water transparency, and so forth. So this is our one of the main ways that we distribute our data to the broader public. At the end of every month, we also generally do kind of a retrospective. So we look back at where all of the sargassum was over the last month, according to the satellite data, and we, how much, where is it, and make some course predictions on where it's going to go. And so, sorry. So this. I say course predictions. This is based on these course scale satellite data, and the predictions will be something on the order of what you see here. You're going to see impacts or a lot of sargassum being in the Eastern Caribbean in the next month or so. So we can give a kind of a regional prognosis on where sargassum may be from these data sets. And I mentioned, or I include this specific image because it shows the early 2023. We had an early inundation event in the Florida Keys in early 2023 it started to get a lot of local media attention, which is not unsurprising. What was surprising was when that ballooned into national and then international media attention. I don't mind speaking to the media. I don't love it, but I'm happy to share what I know, what I do with people. But there's a point in which it becomes static. I don't have training for media, and I was not prepared for this. The other thing I wasn't prepared for was a narrative. I thought narratives were something that politicians deal with, but there was a narrative that popped up, and I mentioned it in my talk already, and that is the narrative of the blob. Okay, so the blob was coming to swallow Florida. You could walk from Florida all the way to Africa without getting your feet wet on Sargassum. And I mean, it became very pervasive, at least in my life. Some friends started calling me Dr. Blob, and I was thinking, I'm going to create a blob vlog, like a la Arrested Development. I don't know, so, like a blob. But this is, I mean, it was really telling that even within an interview, controlling a narrative is hard. So here's Tuan Minh talking to CNN. A bit cheeky, but Tuan Minh does go on to explain why it's not a blob. Again, but I do want to note, this is not a live interview. This is a recorded interview, which means there was someone in the control panel that watched this and put this chyron on the top of the video. Yeah. They missed the message. Okay. So this is, all jokes aside, this is important for a couple of reasons. First, I mean, obviously, it's not a blob. If you were to see Sargassum in the open ocean, it looks something like this. It is exceptionally patchy. And if you paid attention to the color bars that I used earlier, they show something like this. 0.4% is that red, bright, like danger color, which means 99.6% not Sargassum, okay? Now, on the scale of an ocean, even having 0.4% of an ocean covered by something is a ton, but it's not. It doesn't span the entirety of the ocean. The other thing I really want to drive home is that it's patchy not just out in the ocean, but the impacts. So we can say something like the Eastern Caribbean may see a lot of Sargassum in the next month or so, but even if you're on a particular island, you can say you can have a huge inundation on one particular beach, and then a kilometer down the way, absolutely nothing. Okay, so it's not necessarily helpful to give that kind of broad scale, course prediction for Sargassum. So I really want to spend just another moment on this. This is a course resolution data set. It's actually one of the best or highest resolution, course resolution, whatever. But this is one of those data sets that we have that allows us to look at Sargassum on a global scale. And I mentioned there's a 10-kilometer buffer where we can't we cannot develop machine learning algorithms that reliably differentiate Sargassum from water in that nearshore region. So this data set is not really helpful, again, to those areas that might be impacted here, but not there. We do have other satellites, though, that are built for another purpose. They are not made to to monitor the globe daily. They are made to monitor mostly land, but also they cover some of the coastal region. And you can see how much of that, those inland Sargassum features you can see, and how much more clarity you have just in general with this higher resolution data set. And I want to point out too, there's a feature here, and it goes along, often you'll see this if you're in the Caribbean, where there's Sargassum, there's a ton offshore, maybe 5 kilometers from shore, and it just sits there. I mean, it's not the same Sargassum that's sitting there, it's moving, but it will sit there and be just kind of standing offshore for weeks at a time. And it's not until the right winds or the right currents come along, pushes it onshore, and that's when it impacts a certain beach. So again, this type of data set, it is not equipped for that, to identify individual patches and which ones may or may not be impacting a particular beach. And driving this point home, the landmass that you saw there, that was Puerto Rico. This is the marine reserve in southwest Puerto Rico, and this is that course resolution data set. And here is where you can really start to identify individual patches and their trajectory, their potential trajectory. Now, just showing you pretty images where you can see that is different than developing an algorithm, a machine learning approach, or whatever that can differentiate sargassum from boat wakes and clouds. I mentioned some of the other features that can sometimes mask or confuse sargassum detection algorithms. So this is some work that our recently graduated master's student Sully put together. I'm really just going to drive the point home one more time in that on the left side here, you have a MODIS data set. That's that coarse resolution sensor. It's masked for all these inshore waters. So that's the MODIS on the left, the coarse resolution, and the collected on those same days are the high resolution data sets on the right there. This is the NSI, so a high resolution sensor. You notice how much sargassum is inshore that is completely missed by the coarse resolution data set. And this is actually D-scale resolution. They're made to look like they are on the same scale, but actually you can get much more definition even than I'm showing here out of that the MSI detection. We have access to other data sets, not just the one I just showed you, which was MSI, a particular sensor. There are some commercial satellite sensors that we are also using. And a postdoc in our lab developed this approach, which similarly uses machine learning and the planet constellation, the Dove and SuperDove satellites. And in the same way that that Sully did her work, their work, this approach is identifying sargassum in these high resolution products. And what's awesome about this data set, this is 3 to 4 meter spatial resolution. And because there are so many of these satellites out there, we are able to get near daily repeat sampling. So over the course of this week, we have 4 different pictures, and you see sargassum delineations for this very small region of Cancun for each of the individual overpasses. It's worthwhile to note that it's quite dynamic. So you can't just get a picture of sargassum one day and think I've got this for the rest of the week. You really need that high spatial and high temporal frequency in order to monitor sargassum. Now, it's one thing to monitor sargassum, it's quite another to forecast sargassum. And this is some work that we recently got funding from NOAA to do, and Yonggang and many others are contributors in this work, IBO as well. And basically what that involves is coupling our sargassum detection algorithms with hydrodynamic models. So we know where the sargassum is and we know where the water is moving, and we can make some estimations of how the sargassum will move with that parcel of water. So what I'm showing here is essentially that coarse resolution data set. So this is the data that we would currently use for something like the existing SAWS system. And high sargassum abundance is in red and low in blue. But overall, you can see sargassum moving with the surface currents essentially as you would expect. But neither the sargassum detection approaches that we've developed, nor the surface hydrodynamic models that are used here, the AMSES model, have enough resolution to see anything that's happening in this nearer shore coast. If you see that, yeah, it's kind of just stuck. They're not built for that, and that's okay. So what we are doing is trying to couple our new high spatial resolution sargassum detection algorithms with newer or improved hydrodynamic models that are built for resolution in these very near coastal environments. And from that, we hope to be able to make alerts not just for a region. This particular island may experience some sargassum inundation in a week or so, but down to the level of an individual beach, or at least a region of an island. And what that allows managers to do, if they have a couple of days of time, they can stage their equipment to make sure that they're ready to get that stuff out of the off of the beach, or even install some booms or something so that they can collect that sargassum before it becomes a major catastrophic impact. Okay, wrapping all that up. So, sargassum has the duality, both an essential fish habitat and a real menace for many coastal ecosystems throughout the Caribbean and into the Gulf Coast. We have a SAWS system. This is the framework that is currently active, and it provides real-time observations of the sargassum condition. Mostly, it provides retrospectives as well, with some coarse scale predictions. And this product is used quite widely throughout the world, or at least throughout the Atlantic Basin, by many different stakeholders. So although we're very proud of that, we also recognize that in order to have more effective use of our satellite data sets, we need to be moving into this new regime, which is this higher spatial resolution detections, and then coupling with these hydrodynamic models such that we can make inundation forecasts and projections for which areas might be impacted by sargassum and when. And so those are my take homes, as well as this one. There are blobs in the sea, but this is not one of them. Thank you.
Rafter. While I am new to the College of Marine Science, I'm not new to marine science. And so when I was thinking about what to present today, I could have given one simple story, which would have been easier for you to digest, perhaps. But I thought, I've been here 8 months, and not everyone has gotten a chance to really know me yet, or read my CV. And also, I like to collaborate, and I also like to brag a little bit about my work. So that's what I'm going to do here today. So I'm calling this a virtual CV. And so it's going to touch, there will be a couple of vignettes here. I don't expect you to understand all of it. But if there's something that interests you, I do hope you'll reach out to me and say, ask me more about it, or maybe we can work on something together, follow up to that question. So, okay, so I chose this background because I didn't know what to put on such a this is like a personal talk for me. This is not a job talk. You already hired me. And it's not really, I decided it's not going to be a hardcore science talk. So what am I going to do? So I was walking my office and I said, well, why not just throw up some of these books on the bookshelf? I've read a couple of these. Some of these I just have because the covers are cool. David Nahr, you gave me the Oceans book, which I was very happy about. But the I think the book I want to point out here is this Global Environment book by Berner and Berner. This is for a class I had at Florida Tech back in the 90s. And it's where I learned of the term geochemistry. And I think up until that point in my education, I had been an engineering major, but it was boring, to tell you the truth, no offense. I just was I was looking for something else. I was looking at the environmental sciences and oceanography, and I heard, and geochemistry, that term unlocked something in my brain. And here I am, still a geochemist. So yeah, there's we can talk more about these other books if you like, but let's get into the talk. So my virtual CV, I got my PhD from Scripps, and did my postdoc at Princeton. And then there was this postdoc, I call it. It's sort of this soft-ish money researcher position I had at UC Irvine. It was nice because it kept me in science. It was I shouldn't say not so nice. It could have been better, I guess, except that I had a sort of a part-time job as helping to run a radiocarbon lab in these 10 years. And then I did a full scientist workload on top of that. So I'm just glad to just be a scientist now, and teacher and so on. So I've been here at CMS since January. A little bit more about my CV. Some pretty good publications. My first student-led paper came out earlier this year. That was really exciting. I brought in more than $1 million of funding. I've taught 3 years of intro to climate change. I'm going to teach something next semester, probably marine biogeochemistry. And then, of course, mentorship. I like having people in the lab doing fun stuff. So more than 60 undergrads have come through my lab in the past 12 or so years. 4 senior theses. 2 graduate students, both are current. The first is a student I co-advise at UC Santa Cruz, and his name's Ryan Green. He's about to finish his PhD, probably in the spring. And I was very happy. I led the grant in paying for his first 3 years of work. And then, of course, we have CMS's own Alexis Vargas. Alexis, you want to just hi? Yeah. So she's new, even newer than me here, but we've already we've already gone on the road. We went out to work in a radiocarbon lab, learn how to make the measurements, learn how to prepare the samples, and so on. So yeah, there's a little bit about me personally. Now a couple of research vignettes, and feel free to stop me if you have any questions, but also reach out, of course, if you see anything that you find interesting. The first, I guess I'm just sort of going sequentially here. My PhD work, I think, is kind of demonstrative of my approach, though, to science. So the question was, you know, what do we expect to happen to the El Nino Southern Oscillation? That's ENSO, with rising CO2 in the atmosphere. And so while I can't use sediment cores to say anything about individual El Nino events, we could say something about long-term, you know, like 1,000-year-long changes in their frequency. So a bunch of wiggles. I know there's people that can make fun of paleoceanographers, but yeah, I generated this work, and essentially we found no relationship between atmospheric CO2 or even iron. Thank you for pointing that out, Pam. Iron being an important factor for productivity in the ocean. But instead, we found this very strong relationship to incoming changes in seasonal heating instead. So sort of a simpler explanation, and sort of I think is sort of a rare source of good news. Like we're not expecting a change, and so with that, at least on these longer timescales. But it wasn't just this sediment-based work. I also spent at least 2 months out to sea doing sort of ground truthing this process. And so this is my approach is like, okay, so we're going to look at the one thing, but also I'm afraid somebody's going to prove me wrong over here, so I'm going to do that too. And so I did that for my senior thesis. It's led to a whole bunch, or not my senior, my PhD. And so this led to a whole bunch of additional papers. Now, if you didn't follow any of that, or if you got turned off by the wiggles, I understand. I'm always at the end of these vignettes, I'm going to tell you what I want you to understand from what I said. So, and so the El Nino Southern Oscillation, not sensitive to changes in atmospheric CO2, seems to be completely dependent on, at least on these longer timescales, on changes in Earth's orbit, actually. Lucky to receive some funding for that as well to follow up. That's just now passed and has a new paper coming out from that fund. All right, so vignette number 2, I'm going to take a quick hydration break. So sort of a completely different, seemingly different topic, but you know, if you remember my title, it said carbon and climate. And so these are all kind of working together, at least in my mind. So what we're showing here is a model result, so model predictions of surface pressure anomalies during the last glacial maximum. And that'll become relevant in a second here. The question I want to answer here is, so can we reconstruct wind strength, direction, and the wind direction, and therefore have some sense for atmospheric pressure across, you know, known climate change? And so once again, we're looking to the past for some answers, or at least for some testable predictions that we can then take to the modelers or take to or ask some more questions about. So I've been working on a sediment core right there, and actually that arrow, sorry, it's white on the background, but that arrow right there is showing the only direction the winds can go to upwell waters at this location. We just happened, you know, I didn't just happen. Sorry, I deliberately chose this site, so it was between it's downstream of a valley, a gap in the very steep mountains right there. So the winds can only go right into the Gulf of Tehuantepec from this sort of northeast direction. And so we're able to say a couple things about wind variability and strength. I'm working with some actual atmospheric dynamicists on this to get at some, produce some insights, some once again, some testable predictions about these proxy results. And among the things we found are, you know, our understanding of sort of static climate states, such as the last glacial maximum, so colder than today, different boundary conditions, but we have this, at least with this one test of atmospheric pressure distribution at this time, we have a pretty good sense, at least based on this one record. But as we move out, as we warm from the last glacial maximum to the current warm period, in a so-called transient, what we are finding with, I would say, is a fairly robust record of wind strength, or at least wind variability, is quite different than expectations. So that's about all I can say on that. This is really new work. If I shouldn't say if you guys received my all at CMS email, I guess all of you did, and then I learned all of the alumni did as well. Sorry about that. Reason why I'm not saying more about this is because it's very new and it's a new grant that I only just got transferred over here the other day, and I'm still figuring out how to spend it. Anyway,
We'll help you.
thank you. Please don't. Yeah. Please don't spend. Don't use that charge. Oh man, that was. I left the building after that. After I said some very. I think Chris probably heard me across the hallway out what I said. Okay, so third vignette. How much time? Okay. Back to carbon. Okay, how well do we understand the global carbon budget? This is kind of a fun and interesting study. So because I had to change my mind a couple times. There's this existing record back to 2007 that suggested it's very, very old seawater, meaning has very little radiocarbon in it after the last ice age. And so I started a study to prove it wrong, and I ended up proving it right, which made me kind of have to rethink a whole bunch of things about what I think is going on with the global ocean. So next, I said, okay, well, let's look at if it's real, then, and it's in this sort of like well ventilated-ish part of the ocean, it must have some old or geologic carbon added to it. So geologic carbon having no radiocarbon, all of it's decayed away. And so in this study, what I did was, and all these are new measurements. There's an arrow behind these symbols here that goes all the way up the coast. So it's a coastal current, an undercurrent that moves all the way from the eastern tropical North Pacific up to the Gulf of Alaska. And so I said, okay, well, let's create a bunch of these records. Not only once I proved it right, I said, all right, well, let's look at the extent of this feature and see if it was dragged in there, if it was ejected in there, or if it came it was something local. And so if you compare the colors here, so one of the original records is here in orange, which is a very older radiocarbon age, and you can see it's sort of younger, and then it gets old right around here. There's sort of a hot spot of these old radiocarbon sites, and then it sort of diminishes downstream. So this is work we're developing now, but before the, I have an additional study where we propose that it must be this bicarbonate. So and that's because we have carbonate sediments persisting. And just to back you up, give you marine chemistry 101. If you just bubble CO2 into the water, you're going to dramatically lower the pH and dissolve carbonates. And carbonates that is what we are measuring. So we know that didn't happen. So we propose that it's coming from bicarbonate. That's going to come into the story later. But this got a little blurry. This is Ryan's work, so it came out earlier this year. I wrote a proposal, and I said, all right, I don't really want to do that much more of this work. I'm kind of getting too into this rabbit hole, but I do want to make sure someone else doesn't come along and prove me wrong. So let's put some numbers on this. And so we brought Ryan in to do that. And so first we look at these sort of global scale impacts. And these are just, this is the time periods and the volume of different kind of carbon that could be added to the waters. We found that, once again, it kind of supported what we originally thought. If it's bicarbonate, we could add a lot of it. The next chapter in Ryan's work that we're editing right now is really focused on this not the global signal, but the regional signal. And just a very quick background, this region is somewhat popularly known as the shadow zone. So the physical oceanographers will understand there's a potential vorticity barrier at sort of these upper intermediate depths that allows prevents a lot of these lower latitude sites on the eastern Pacific from getting well ventilated, more oxygen brought in. So they're sort of considered this a shadow zone. And so now we're kind of looking at a new model that we worked up, some new calculations to actually constrain what is happening to this shadow zone over these time periods. When we have this understanding that the signals are real and that it must be this one form of carbon. Now let's put some constraints and once again some testable predictions on what's happening to these waters over this time scale. So we're looking at a range of mixing rates and sort of the volume of that shadow zone. This is all constrained versus the Gulf of California, which we're assuming is a fixed volume. And so we're able to get these sort of plausible numbers. Okay. And so we got a couple, NSF like this. Maybe I should keep going down this rabbit hole because NSF really, they seem to like this. Here's the kind of TLDR. Geologic carbon supply could be pretty large. We're looking at less than 850 petagrams of carbon potentially added. But it's kind of sneaky in that you can add, if you're adding it as bicarbonate, you're having sort of a negligible impact on atmospheric CO2 and even the isotopes. We're looking at the C13 value of this carbon. If it's coming from carbonate, it's not that different than the ocean. So it's sort of sneaky, what I like to say. And so this is my transition to, this is sort of, I was talking about, the past and present, my research into the past and the present. Now let's start looking at the future. And the future for me is CO2 chemistry, I think. Now I'll be pursuing and persisting with a few of these other projects. But one of my big inspirations right now is to move into this space that, to be honest, I tried to give this idea to someone else and no one would take it. Now, I don't think that means it's a bad idea. I think it means they didn't get it. So I'm still doing it. And you're about to learn what that idea is. But first, introducing marine carbon dioxide removal, or MCDR. You may have a thought right away. We should do it or we shouldn't do it. That may be a thought you have. That's totally reasonable thought to have. Personally, I think that as oceanographers, we need to be in this space, and that a lot of us here, probably all of us, can contribute in some way, right? So, there's going to be impacts from this marine carbon dioxide removal, even tests that we don't know what the impacts are going to be on the fish, the larvae, and so on. Physical oceanographers, we need help in there for sure. Geological oceanographers, for sure. There's a lot of need for our expertise. And I don't have time to tell an anecdote, but it involves a private equity firm, private equity dude who asked me to review a proposal on marine CDR from a Google dude. And it was so Looney Tunes crazy. It made me go into this world. Like, we need experts. So there's so like, just very quickly, how are we going to do this if we're going to do it, if we decide to do it? There's this economic model that's in place for rapidly scaling up marine CDR. And it involves a variety of things and I'll walk you through in a second. But the thing that I think most of us can contribute to is this, or at least chemical oceanographers can contribute to, is this measurement, reporting, and verification. A lot of acronyms. I blame NOAA for being involved in this space. But for example, let's just say you have some marine CDR activity. I'll call it an attempt because we don't know if it's been successful until we measure it. And so, for example, adding alkalinity to the water to increase the ocean's ability to take up CO2. Then this must be followed by MRV. So some kind of ground truthing of what happened. Some people call it additionality. There will be models involved. But personally, I think we cannot only rely on models. We're going to have to ground truth those models. Next, there's a carbon credit that will be supplied. It can be either given to the person or the company who did the marine CDR or so on. There's some money to be exchanged there. And then that will feed into more investment of marine CDR. This is meant to motivate the case for this measurement reporting and verification. Okay. And so this is where many of us can make a contribution. So what am I doing? This is the idea I tried to give away to modelers and different friends working in this space. And yeah, it's the idea is to use isotopes. I mean, I'm an isotope geochemist. It's a little bit like, you know, I have this hammer, everything looks like a nail, but for real, I think it will work. And so what we did was, before we've done any measurements on going out to sea, let's do some modeling work. So once again, teaming up with my friends at Santa Cruz, and actually my student at Santa Cruz, I turned him completely out of paleoclimate and paleoceanography, and now his third chapter is entirely this. And he's gonna, I don't know if what I did was good, but he's gonna quit science. He's gonna work for a private, you know, marine CDR company, and maybe it's better for the planet. We'll find out. But yeah, so we did some modeling work, setting up my lab now to make seawater C13 measurements, high quality, high volume, high throughput. And for example, in the old days, it would take you basically 2 days from sampling, you know, getting your seawater to measure. 2 days later, you might have a measurement. Now we're 15 minutes. So this is really exciting. So that's what I'm doing in one of my labs. The outstanding question here is how to relate the observations to the models. So we're going to need models. We're going to need observations. We don't want to make a million observations. We want to make just enough to make the models good, to do this MRV. And so we're seeking money for that. So that'll be going back out to NSF ChemOsh in February. All right, so I'm counting down. Is that 3 minutes left total or 3 minutes to? Oh, okay.
Well, in 25 minutes.
In 25. Okay. So I didn't even touch on everything because like I said, I'm not new to marine science. So, but if I'm throwing this up here not just to brag. I'm hoping people will be interested and want to collaborate. So large scale marine nitrogen, iron, and carbon cycling. Thank you to Tim for my sole author, Rafter 2024, for that offer to write that paper. I'm very interested in the physics of upwelling and the surface mix layer variability. We have these proxy results and modern seawater biogeochemical results that I think are, I'll just say interesting. I think they're very interesting, and for people involved and interested in upwelling and how you deliver chemicals to the surface ocean, very interesting. We're also finding some very interesting ocean climate dynamics with the mix layer variability in the Eastern Equatorial Pacific. This may seem to come out of totally left field, but it's interannual and decadal modes of ocean climate variability. This was actually based on a coral record I worked up. So this is El Nino, this is like actual measuring El Nino events, Pacific decadal oscillation, and so on. Last but not least is a paper very dear to my heart, looking at global ocean overturning, ventilation, and air sea CO2 flux on a variety of timescales. So if you wanted to download this talk for any reason, there's a QR code, and I'm here to answer any questions that you have. Thank you.
I'm Chad Lembke. I work at COT. I am not a faculty member. I am a support staff. I'm an engineer. I am not a scientist, and I like to say that because especially here. I also do not have a PhD, although I let P-card people think I do, so they may answer the questions a little faster. But this is going to be, I feel kind of bad because a couple of people have asked me about the title, and it was really just because I was afraid Sean Min was going to make something up for me. So I'm pretty much going to talk about the things I talk about, but it is a little bit of, we do a lot of making do with what we have in order just to get the job done. And there's a misnomer a lot of times that gumbo is just throwing everything in a pot and making something that tastes good. It's actually much more than that. And I think a lot of times in science, we make that mistake of just thinking it's going to happen. So I'll talk just very briefly about my role and the other engineers and technical support staff in the college. I do a little bit more than I intended on the glider program that I coordinate with a great team, and the COMET project that many of you are aware of, a mapping center for NOAA, and then some SBIRs that we've gotten for some optical sensing with a local company. So we've been a group since '95. I joined in '98. At one point, we grew up to over 80 engineers and technicians for a while. That's one of the t-shirts that we had for our Oktoberfest that we used to do every year. We are now down to somewhere between 6 and 10 of us or so. The thing that new students I'd like to emphasize the most is that we have a lot of gear. We have a lot of leftover gear. A lot of the leftover gear from previous projects finds their way down to us. That can often be just recalibrated and used for your projects. Okay, so please, if you have a need for field equipment, a lot of times, or lab equipment, a lot of it is kind of old, but we may have something laying around that could be of help. We also have a lot of historical knowledge, so just stop by and ask questions, and we're happy to help. Jumping right into the gliders. I assume most of you understand what gliders are at this point. They've been around for a while. They're especially useful in coastal regime for getting samples from the surface to the bottom. We work with a lot of different people. We get funding from a lot of different people. We run them primarily in the Eastern Gulf of Mexico and the South Atlantic Bight. Like I said, we've been going since 2009. The graph on the top is glider days per year. So last year we had over 450 glider days in the year. That a glider day is a glider in the water for the day. That's just kind of a common metric for going through and measuring your how busy you are. This year we will not get quite as high as last year, but you can see the footprint of where the vast majority of our deployments are. All of our gliders have CTDs and fluorometers and dissolved oxygen sensors. We have one that has a Suna nitrate sensor that I'll talk about. Most of them have passive acoustic monitoring. We almost always put a tag telemetry receiver on them, and one of them now has a water column echo sounder on it. Other things to note is that all that historical data that we have is available since 2009. So any of those areas that you see, we have that data, you're welcome to it. And we frequently have trips of opportunity on charter boats. So for people that would like to get out of the office for the day, please let me know. And we regularly take students out. There's almost always space on the boat. So what do we do? In the all those funding agencies and other universities that we work with, we work on helping a large group here with red tide prediction. We do a little bit with extrapolating the satellite imagery from the surface down below. We do some work in augmenting circulation models. We have tried to venture into more of the counting and tracking fish, and in conjunction with that, water column biomass. Recently, there's been an effort to get oceanographic heat content for hurricane strength forecasting. And then some of the newer things that we've been doing are sound speed for mapping purposes and even gross bathymetry maps. We did do a little bit during the oil spill back in 2010 as well. So red tide tracking and forecasting. We work with Sean Min and Kristen Buck and Bob Weisberg and Yang Gang, Maya, and the folks next door at FWC. It's a holistic, kind of systematic approach to understanding the entire environment and how the various variables can impact whether red tide is going to bloom or not. The glider portion of it is purely getting subsurface data on a regular basis, pretty much from in the Tampa Bay region. You see in this image here, the circles are cruise stations that for Chris and Buck's nutrient cruises. We fly the glider along that path back and forth all year long, pretty much providing variability as to what the water column is doing and how it's changing over that time period. That's been going since about 2000. And that data is used to help understand circulation and then again extrapolate the surface environment down to the bottom. One of the areas that we've focused on is adding nitrate to the glider. So we have a SUNA nitrate sensor, which is a low resolution nitrate sensor. It's a little finicky. It's not the best sensor in the world. So what we do is we compare it to the cruise data whenever the cruises go through, but we've gotten pretty reliable results. And what you'll get is a transect from near shore to offshore. And as the year goes along, the nitrate picks up at various depths based on the seasons and the upwelling and whatnot. And so while we're doing that, we also get temperature and salinity and fluorescence and compare that together. We, of course, don't do the analysis. That's more Sean Men's group and Bob's group and Kristen's group. But you can start seeing patterns over periods of time, which is what gliders are really good at when they're used in a sustained and repeated way. So moving on to echo sounders. The idea being trying to get water column biomass on a regular basis. So you saw this image right here. This is nitrate from the surface to the bottom as it goes up and down. We would like to get the same thing with water column biomass as much as possible. Or at least that's our hope that we'll be able to get there. So an echo sounder on the glider is a relatively power hungry instrument, but we get to see layers of everything from plankton all the way down to fish, or up to fish size. So when we get them at the same time as everything else that I've mentioned, CTD, dissolved oxygen, etc. It's a relatively large data set, so we've done a lot of work. We had serial number 1 integrated into a glider, both 2 different manufacturers. Working with NOAA to do that integration. Part of that is the difficulty of that is that they are relatively time consuming and comprehensive data sets to analyze. Getting that back real time over satellite takes a heck of a lot of data compression. So we've developed some algorithms to take real time data, send it back through the satellite, which you can see on the left, which actually looks like when you first get it off when you get the glider back and you get the full resolution data, it looks like the center, and then you have to calibrate it and correct it for depth so that this is the bottom, this is the bottom. But we get these for long periods of time over and over, and so then if you do that for a month, you can start compressing that. The top right is acoustic backscatter for 2 months on the West Florida shelf, going out to the shelf break and back. Analysis of that done by FIU. The size of the circles is the amount of acoustic biomass in during that profile. So you get maps of how many fish or how many whatever you're looking for based on the acoustic biomass or acoustic signature. And then at the same time, you get the chlorophyll and all the other good stuff. And so you can start comparing those. We'd like to combine that as well with passive acoustics. So just listening. Passive acoustic monitoring, you can listen for anything that makes sounds, whether it's anthropogenic noises or fish. Here we've done a lot of research with red grouper. Red grouper make a very distinctive sound. You can easily process that out. We've been doing that since 2012. Former faculty member here, Dr. David Mann, and then one of his students, we published a fair bit, and then we've been doing it since then. So we have, I think, about 50 missions on the West Florida Shelf and South Atlantic that have passive acoustics. We are now starting to look at Rice's Whales calls. That deployment that I just showed, this one right here, where we went along the shelf break, that goes up into the Rice's Whales main domain. You listen for them, we heard between 200 and 300 calls during that time. We've had analyzed by 2 different people. The folks at NOAA Southeast Fisheries Science Center, Melissa Soldevilla and her team, as well as Bob Courier at GCUS, is developing an auto recognition program. So you combine the biomass, which is generally possibly the food for the Rice's whales, combine that with actually detecting them. You can start setting up a real-time monitoring system like it's done. Some of this is done similarly on the East Coast for right whales, just with the passive acoustics, not with the echo sounder data. Another thing we do with the gliders is heat content. That's been a national effort from IUs and ONR to collect data all through the Caribbean, Gulf of Mexico, and along the Atlantic coast. We deploy gliders year round, throughout the hurricane season. And that data is ingested into the national models. What's shown on the bottom is just simply, this is the day that the glider was deployed. The glider data is in the blue, 3 different models. You can see how poorly they line up. Within 5 days, they line up a fair bit better. So it's getting ingested into the national models, the RTOS model and GOFS models, as well as the European model. And it straightens it out relatively well, or helps it a fair bit. We did have 2 gliders out during Adalia. One of them was lost during that. That's the first time we've lost one. But right after Adalia came through, we put another one out. So we had data before and after Adalia. We saw a significant temperature drop in the water. We had one offshore off the shelf and one inshore at about 50 meters of water. The ones at 50 meters of water, there was a wave buoy within 20 miles of it that was recording 10-foot waves, 10 meter waves in 50 meters of water. And that's when it broke and washed up in Pensacola a few days later. If you are interested in our data when they're live, when the gliders are out, there's at least 3 different sources. One of them is our own here at USF. Garrett Miller designed and built the brewery, which is a nice real-time monitoring system. It gets us the sensor data as well as the flight data from the glider. We put one out yesterday. We picked one up and put one out. Heather Broadbent and Randy Russell went out and swapped them out. And that glider that's out now has an echo sounder on it. So the echo sounder data that's coming back real time, you'd be able to see it on the brewery. GCUS is another place where we publish almost all of our Gulf of Mexico data. They usually take our Atlantic data as well. And then all of it gets ingested up into the national archives at IUs. Move on to mapping, body mapping. I think we all understand what mapping is. This is a relatively large team. I think that's most of them. That does not include all the students involved in the project. It's it's a dynamic project in that it has a lot of physics. It has a lot of the mapping portions of it as well. Our portion of it in COT is working on revamping a surface vehicle that Dr. Chris Kanowski revamped for us. They named it after the possum that lives under KRC. And then re-envisioned is the possum in in a more AI generated way. So it's a test robotics environment vehicle for ocean research. It works with, like I said, Dr. Kronowski got it going with a student who was a student, Jaden Crute. She is now a full-time staff member for now with us here. Matt Hallmeyer and Alex Silverman is also working on it. This is what it looks like. It was refurbished from an old vehicle that we had back in the mid 2000s, and it was used for water quality monitoring. Chris came in and pulled the guts out, and we reused some portions of it, but the vast majority of it got reworked, and we re-engineered the controls and software and the navigation and power supply. Jaden built some new 3D printed propellers that we're testing hopefully next week. We were trying to test them this week, couldn't get to it. But all of this is utilizing open source data with the robot operating system code that is, I think, primarily developed at UNH at their mapping center and in collaboration with Chris. And so we intend to continue using this and trying to get it more accessible to more students if there are any that would like to get involved. This is one of his first field testing deployments up in Georgia that Chris ran. It, like I said, it runs on waypoint navigation and or hand control, like with a regular old joystick. You can drive it around, or it has a full programmable navigation system. So far, we've integrated a Kongsberg EM2040 sonar, as well as a handful of cameras and other sensors. We're working on continuing to expand it. Again, we're going to continue writing this in open source code, hopefully, so that the idea being that anything that we do, we share with other groups, and they will be able to utilize. And similarly, there's a lot of code out there that we can grab and hopefully retrofit into this. So I think we're a little bit of a test case in trying to do that with an ASV. This is a direction that Chris and the folks at UNH kind of pushed, and so we would like to further that. Just, you know, the cutting edge of surface vehicles is being done and is being done other places. We really want to make this more of a teaching tool for either the College of Engineering and or here. But with that sonar, we are able to do some maps right here. That's on the corner of the peninsula. So that's the dark beige is MSL. So it's right down in the corner there. We just did a little test pattern of about half a dozen lines, collecting bathymetry data. Next, briefly on the COMET project, we have a backscattering reference site. If you're not familiar with backscatter on multi-beam and sonars in general, the backscatter is the strength of the return, which can indicate the bottom composition on some level. The difficulty of it is calibrating it and knowing what that strength of return means. And so we're going to be setting up several offshore locations that will be reference sites for multi-beam backscatter or echo sounder backscatter. We hope to have a field campaign in a couple months here in this water shelf. And I believe that if you're in the geophysical tools class, you may be coming along.
Counting on it.
Other thing we're playing with is exploring some visualization techniques. This is a bathymetry map that was collected offshore by Matt Hallmeyer, and it's a sinkhole about 10 miles offshore, I think. And this is just the data. The process of, you know, the user interface for this is allowing the user to see it in 3 dimensionals and move around in it in 3 dimensionals. This is a bathymetry map. It doesn't have to be a bathymetry map. It could be a circulation model. It could be any 3D point cloud to be able to look around. And it really is, just looking at it when you watch this here, it looks kind of trivial. When you're actually in the middle of it, it really does kind of change the way you see what the things are around you. You see things just a little bit different. It's a very, very nice guest performance by his daughter Ellen. And please note that I do have names on most of the bottom of these. I have pictures on the bottom as well. Feel free to reach out. One last thing on COMET is a combination of bringing the gliders into it as well. Sean Beckwith, one of our students, has developed a bathymetry, glider-derived bathymetry product that has used all of our historical data. So this is an area offshore, about 90 miles offshore, where we have a lot of glider tracks that he went through and created an algorithm to. And we also have multi-beam maps of this area. He developed an algorithm to derive the bathymetry from it, taking all of the altitude adjustments of the glider back out and putting it and creating a map out of it from the altimetry data. And then just coming along and looking at the difference between the multi-beam map and his map. He'll be talking about it whenever he depends. Last thing I will talk about briefly is a SBIR project we have with a local company with Dr. Scott Sampson. He's a former COT employee from way back in the day, and he's an optical engineer, and we're trying some new time of flight sensors that the whole world of optics, especially with the advent of autonomous vehicles, has really changed a little bit. And it's finally gotten, a lot of the sensors that we use in oceanography, optical sensors, are based on 1980s and 70s technology. This is finally a step forward in the processing and capabilities of some of these sensors that could really be much more sensitive and much faster and allow you to do some things that are really good. So we've been working with Dr. Hu's lab, as well as now we have an engineering student helping us out, and we're testing in two different ways. There's looking at trying to do cheaper and faster and more sensitive, but also trying to do things at a distance. And we've had success at both, enough so that we've got a phase 2 SBIR that will be starting here in September. And we would be looking, we have our first prototype ready to put in the water, but we will be looking for possibly other people to partner with, because at this point we're just doing chlorophyll fluorescence. We picked that because it's easy and there's lots of sensors around. We do believe there are lots of other things that we could do with this. So, and last thing I'll just briefly mention is Dr. Mrowski's new vessel that is available for the college. It should be up and running by next week or the week after, I think. So feel free to stop by and talk to Steve or Cheryl anytime you might want to. It's 35 foot vessel. It runs at about 35 knots. I'll leave it there. Thanks.
So I'm going to talk about something that's only about a year old. It's this expedition that we conducted 2 summers ago. That was 5 years in planning, and I've titled it here after an infamous paper from back in the 90s, What Happens to Terrestrial Organic Matter in the Ocean, which is sort of the crux of the proposal that got this expedition funded. Knowing that I'm the only thing between you and beer, I did you the service of numbering the slides and back. That'll just count down, and then when we get to 0, we can go to the happy hour. This was the group of folks that was involved in this expedition, both on board the ship with my co-PI Weiyi Gali, who's at Woods Hole, and we had a bunch of people that were shore-based scientists as well, ranging from folks at the Brazilian analog to NASA, who are sending us satellite data so we could decide on some of our sampling stations, as well as folks who are just waiting for samples to come into the lab and to measure. So this, all of these people, of course, are responsible for everything you see that's good, and anything that you take issue with, that, of course, is me. To get started, this is a basic carbon cycle diagram of Earth. In blue are the size of reservoirs, and in red are some of the fluxes. And oftentimes when students look at a diagram like this, you look at the big numbers, right? And what I want to talk about mostly is this the one that works?
It's the laser.
That's a laser. Oh yeah, these big numbers like the Earth's crust, and of course we're interested in what's in the atmosphere. But the number I want to really talk about is this little tiny number here. That's what makes it into marine sediments. And people often overlook that because it's such a small number, but it's an important one. And in fact, over geologic timescales, small changes in that flux control things that are as trivial as the composition of our atmosphere, how much oxygen is in it, how much CO2 is in it, right? So even though it's small, it's mighty. And that's what this proposal was focused on, especially as we get into the second century of the largest geophysical experiment that we've ever undertaken, whether we authorize it or not. And here, of course, is that iconic question I mentioned. This was John Hedges at the University of Washington in 1997. What happens to terrestrial organic matter in the ocean? You would think that we know this by now, but it's still a raging question. And I like the way he puts it in this abstract, that either our global budgets and distribution estimates are greatly in error, or both dissolved and particulate carbon of terrestrial origin suffer rapid and remarkably extensive remineralization at sea. So think about that second part. That means it's stuff that has come off of land and has already been used in some respect by biology and is degraded, enters the ocean. Why would it then be favorably used by the ocean microbial loop and other organisms? So to get at this question, there are basically two end members of river transport into the continental shelf and into the ocean. And one is like the Ganges Brahmaputra here, which is draining the Himalaya mountains. And this one has so much sediment dumping in the ocean that it actually pushes the crust down and creates more accommodation space and rapidly buries the organic matter. So a lot of this, the terrestrial organic matter, when it gets to the ocean, we know that it gets mildly degraded, but it also gets rapidly buried and preserved. So that's one end member. The other end member is the Amazon River, and both of these are major systems with a large proportion of the sediment that's coming into the world's oceans. The Amazon River has a subaqueous delta, but a lot of the mud and water actually gets entrained by physical oceanography currents and waves from the trade winds and equatorial countercurrent, and moves up the coast in a belt of mobile mud called the Guyana's Mud Bank. Okay, so this is the other end of the spectrum, and it has an opposite effect on organic carbon coming from land. In fact, because this is constantly getting stirred up and reoxygenated, the thought that this is sort of like an incinerator, and it just burns up all of the terrestrial organic matter, rapidly replacing it with marine organic matter on the coast. Okay. And a little bit more background here. We have this is a typical continent. This gets us into talking about carbon isotopes and radiocarbon, which is one of the tools that we want to use to look at these sediments. If you take a continent at its highest point, at the continental divide, a drop of water that lands on this divide and moves down, eroding material with it, will include some carbon. That carbon is generally given by this black color. It's kerogen, or really old rock derived carbon. As it moves down on an active margin, which goes into the ocean with a narrow shelf and into deep water, it mixes in with modern carbon from soils and plants. It gets a little bit of marine carbon, and then it gets buried in this form. Whereas on the passive margin side, there are usually large, vast floodplains, and there's lots of erosion depositional cycles where the sediment can kind of get held up on its way to the ocean. And that changes it into this modern plus aged terrestrial. carbon, and then that goes into a broad continental shelf and gets replaced with a lot of marine organic matter. So this is just another way to look at those two end members. So as I said, this question isn't new. People have been trying to figure it out and using the Amazon as one of their end members for years. There have been several expeditions to the Amazon. You'll notice that all of these are on fairly large ships. The Columbus Island visited a few times, more recently the RV Nor. And because of these mud banks, it's not advisable to take a large ship close to the coast, which inhibits these expeditions' ability to get into what actually is the mud bank. So they're generally studying the shelf edge and outward. We wanted to get in a little closer than that. And again, this is going back to the mid-90s. I'm going to be working in the space of C14 and C13. This paper thanks the Columbus Island for its efforts. So these two isotopes are useful because we can kind of work in this space of stable carbon isotopes and radiocarbon, which is decaying through time. We can set up mixing models, which this is basically just a weighted average of two fractions that equal 1 times the terrestrial and the marine end member is getting what you end up with in the sediment. So we're going to try to, people have been doing this for decades now, trying to figure out what that terrestrial end member is. What we notice is going back through the literature, if we work again in this space, I flip these axes, this is stable carbon, this is radiocarbon, is that when the first expedition was out there, and the first modern expedition in 1983 that tried to apply isotopes to the work, they came up with this sort of mixing model where marine organic carbon is here in this space, and this was riverine and suspended sediments. And it showed that the radiocarbon didn't have much resolving power, that the main differences were with along the x-axis in stable carbon isotopes. And as a result, carrying forward as the development of AMS technology and more radiocarbon measurements became available, not many people used it until a decade later. At this point, it seemed like the terrestrial end member had changed, but still people were saying we can't really use this. And a decade later than that, we're seeing the end member down here. What's happening here is that this curve is the atmospheric radiocarbon bomb curve. So that was, we altered the natural distribution of C14 in the atmosphere by nuclear bomb testing in the 1950s. And then we stopped. And now we see a decay down. So the atmosphere looks like this. And that's what's making up a lot of the terrestrial end member. So all of these measurements, the red point is here, the orange point is here, and the green cloud is sampled in here. So we were basically coming down that slope. And as a result, it inhibited the use of this dual isotope approach to some extent. And those authors realized that. They said that this line is probably flat, mainly due to the thermonuclear bomb testing. So we found ourselves in a position not only in time where we could use this isotopic system in more to more efficacy, but also having developed some techniques to use, because right now we've noticed that shelf sediments along this mud banks transect, they don't sit on that mixing line between the two end members. So my first PhD student back at Tulane University applied Pyrox C14 analysis, where we gently heat a sample to 1,000 degrees Celsius in helium with no oxygen. We break it apart. We then oxidize it and take that carbon and measure it for its isotopic composition. And we found that old terrigenous organic carbon actually persisted way downstream of the Amazon River along the French Guiana coast. And this was sort of a surprise, because we'd always thought that this was an incinerator and that it would get rid of that terrestrial carbon very quickly. So these are the sites that we analyze. The Amazon would be down here somewhere. There we are on South America. These were just legacy samples that a collaborator, Mead Allison, made available to me when I was interested in it. And this is what this technique looks like. We heat up the sample on this temperature axis. We get some CO2 off, and then it dies down. The reaction dies down. We can break that into different intervals and measure its C14 age. You can see that we have quite a bit of old carbon in these sediments, and we can quantify that using this method. So the goal here is then to apply these new methods to see what's happening with these shelf sediments, why they're hanging out off of this mixing line, where the mixing line exactly is, using both new techniques, and we also wanted to use a small draft ship to get it closer to the coast. So from this very dynamic environment, we wanted to use the Walton Smith. It was obvious from the reviews that we got on the proposal that this was a pretty good fight between the male reviews and the panelists of whether we could conduct this expedition on a ship. This would be the furthest it had ever gone from its home port, Miami, bringing it all the way down to the Amazon River. So we were funded in late 2019. We planned our field work in that first year, and of course that didn't work out too well, did it? As we all know. So this is a map from the New York Times of COVID deaths starting in 2020 when we were supposed to do our cruise. We obviously, you have to reserve the ship, and you have to make these decisions to cancel your cruise several months in advance. So our original plan was in June 27th, 2020. We were about right here when we had to decide let's not do this. And of course, UNOLS decided to not let any ships out of ports after the end of June. So we could have gotten out. We might not have gotten back into port. So that would have been good. We then planned it for the following summer. But at this point in the pandemic, we had to make our decision, and we weren't sure if vaccines were going to be available, especially in Brazil, where we would have to have a riverboat pilot on board the ship and taking in our other scientists from Brazil. So we decided to cancel it here. And you'll notice now you're seeing a pattern that we're actually pretty good at forecasting when the low points were going to be. Now this was a line that passed. We had no idea, but the U.S. Coast Guard then prohibits U.S. and our academic research fleet vessels from getting within 12 nautical miles of Brazil. And of course, we were going to go up the river with the Walton Smith. So that kind of changed our plans. Then we had a mid-life engine refit happen here during the next time that we were going to go out. To decide maybe we can do this in the fall. And note the engine refit took too long, but you can still see that we're very good at forecasting these lows in the pandemic. So we finally, fifth time is a charm. That's what they always say, right? And we've got it out we planned it for May, and even that got delayed until June of 2023. So the bottom line is next time there's a pandemic, ask me and Valier, or give us money to do a cruise, I guess, first, and then we'll plan it when the pandemic's going to be pretty good and go about your lives. So we ended up having to do 2 ship operations. The Amazon River had to be sampled with a non-American ship. We ran a ship out of Macapá with a skeleton crew of ours aboard. We looked into several options. Bottom line is nothing would work except using a small boat out of Macapá. One thing we learned in this process is that this area is actually further in terms of ship transit days from Rio de Janeiro than it is from Miami by about 50%. That's how big Brazil is. So this is what it looked like aboard the Mirage, which we ran out of Macapá, which is the last major city on the Amazon River. It's about 500,000 people, so it is a pretty big city. But once you get past that downstream, you find much smaller settlements and things. I was not on this leg because I was still trying to get the Walton Smith out. This was basically Weiyi and his graduate student. And then our plan was they were going to take samples, they were going to freeze them at a local college in Macapá. I was going to fly down, and we were going to then drive those samples to French Guiana. And this is what that looked like. These are our samples in a Hilux pickup truck that had 350,000 miles on it. This is the road to French Guiana. This is the state of Amapá, Brazil. The only road out of that state is this, because everything goes by river. So the only way out of Amapá by road is to France, believe it or not. And between these 2 truck diagrams, that's the area of the road that took about 80% of the voyage, because that's where there's no pavement, and that's where it looks like this. The rest of it's actually not bad. So, so this all went very smoothly, just as we planned from the beginning, I guess. Then we got to this bridge, this lovely bridge in the middle of nowhere that goes to France, because it's going to France, so it's big and unnecessary, but it was a very welcome sight. until they turned us away at the border. I'll just whip right through these stories because you can ask me more over beer. This is me and Valier's graduate student making an illegal river crossing with life jackets because, again, it's France. We had to get paperwork filled out by some local official because they wouldn't accept Paris's paperwork. That was after a night of not much sleep trying to deal with the U.S. State Department on a Friday before the border closed on a Saturday at noon, and it was stressful. And that's all of our gear and samples in the back of the van once we got into France finally. So it all worked out, and believe it or not, we had a successful expedition. We did have to get reauthorized 2 times the sample in Brazil by their naval lieutenant that they sent on the boat that spent most of the time throwing up, but also spent a lot of time stopping us from working. We did lose 2 engines, but who's counting? Because we were still able to sample 2.5 tons of water, filter most of that, and also visited 21 out of 21 stations. We got about 875,000 grams of sediment. Lots of measurements of reflectance. It was a good job in only 10 sea days, which were rushed and limping along 1 engine. This is what it looks like. We had a full range of salinity from full oceanic strength to basically 0, which was 0 from surface to the bottom, 90 miles from the mouth of the Amazon. It was pretty incredible. It's what looked like chocolate milk and what the color of my slides represents. And this is up a little bit further. There's the Amazon. Here's off of French Guiana. So we had a pretty big gamut of conditions in water chemistry. CTD data, a funny story is that CTDs are made to turn on when it senses salt water. When you put them in fresh water, you get data that look like this. Okay, so not very good when it doesn't have its pumps turned on to circulate water throughout it. This is what it looks like when we're deeper into saltier water. Deck unit works. Deck unit pump will not start. All right, so these are some of our long track observations. Of course, you see some interesting frontal boundaries. As you're here, we recorded this one. We plotted on the map with this square. You can see that it's right along this boundary in both chlorophyll A and CDOM as measured from the ship's flow-through system. What do we get from these 2.5 metric tons of water? This is still in process. We're rushing through these analyses as we speak. We took total suspended solids, which we'll measure for the C14 to get back to those plots that I showed you earlier. For carbon, we're doing a gamut of isotopic work on different dissolved species. We have a student at USC working on vitamins. It's the first time that B vitamins and retinol concentration have been measured over such a large range of salinity. So he's really excited about that. And then we've done some incubations on board with Amazon sediment from the river. This is what we were trying to keep frozen on our long drive up to a closed border crossing. But it still worked out. We were able to confirm that there's translocation of isotope lignin in these sediments when we mix it with full strength seawater. So that was a fun but still developing story. Nutrients, of course, the Amazon is a source of nitrogen, and we see this diminishing with our transect upwards. I wanted to put this plot here. This is some earlier data from Subramaniam et al. In 2007. This is offshore further. This is where most ships visit this area. And you can see that this is way lower than everything that we were measuring along the coast here. But it's to also point out, kind of relating to what Brian was talking about with the Sargassum. A lot of people have been wondering, has there been an increase in nutrients in the Amazon? And you have to remember that the Amazon, even though it's such a huge source of nutrients and sediment and water, it's relatively pristine. So there's we don't think that there's a huge increase in nutrients through time. We don't have those data, but you can see that it does get an order of magnitude lower by the time you get into what you would call the open ocean, before you get into the Sargassum seeding area. But some interesting things are coming out here. We're not able to sample core waters, but we're able to sample the water off the top of the cores. And you can see here, this is a phosphorus, again, high at the river, lower up here. This is the water at the top of multi-cores. And it's almost an order of magnitude higher there. So this might mean that there's sort of a delayed release of phosphorus from the sediments rather than from water itself into the water column. Sediment coring, we had almost as many pages in our cruise plan dedicated to this as we did for COVID. because sediment coring in this area, you can imagine that there's almost like no sediment water interface. It just gradually gets muddier and muddier until all of a sudden you realize, hey, I'm in sediment. So that means that coring devices don't often work well. So we had contingency plan after contingency plan of what to core. And then we got down there and both the gravity cores and the multi-core system worked like a charm. We just got a lot of great sediment. Gravity coring was more difficult than we planned, only because we kept hitting a thick clay layer about 2 meters under that was gray. It was not the typical chocolate milk color, but this gray color. Actually did some radiocarbon dating on that to see if it was the same age all the way up the coast. But we do see that it's actually older near the river, and then there's this one outlier. It generally gets younger as you go up the coast. Delta C13 and nitrogen 15 both get heavier as you go up the coast. That makes sense because you're going from more terrigenous to more marine influence. These are some data from multi-cores. It looks like just a data cloud. But you can see if you look at these color trends, this is close to the Amazon, this is further, and you can see it's getting heavier, and it's getting heavier and then bounces back because on the outer stations a little different. That was nitrogen. This is C13. Something that's interesting here, we kind of expected all of our sediment cores to look like this square one, 10 MC, because we expected these sediments to be so well mixed. We weren't expecting to see changes like that, for instance, in 17 MC. But we noticed a difference here. You can see this core has actually some structure, which we weren't expecting. This is a CT scan. And you can see that this one comes from that highly variable Delta C13 core, whereas what we expected to see also matched what we expected to see with the sediment. So this and this were a surprise, and they were related. We're still doing some of the analyses. Thanks to the MEC lab here, which hopefully many of the new students have been able to visit. We were able to go through more than 500 samples in the last year, and actually most of that work was over 3 months. And it was helped by students Teresa King and Ryan Schomber in my lab to prep those samples. So thanks to all those folks, including Devin and Ethan, who work in the MEC lab. So we're still going on through some of this work, and what my lab specializes back in this RPO C14. This is the last few slides. These are actually real numbers. I didn't have 70 slides. This is output from riverine sediments. These are the fractions because we can split the samples into different fractions, and you can see that we get a range of fraction moderns from what we would expect in the atmosphere now, all the way to very old stuff that you would expect from the Andes Mountains or something. And there's a slight change in Delta C13 as well. If you measured bulk samples, just the whole sample without doing RPO, you'd be somewhere in that circle, right? So this kind of provides a prism, aptly with this color bar, to see a whole rainbow of different isotope content when we run it through this system. We can add to this our marine end member, right? And these are the samples from my first PhD student, Elizabeth Williams. You can see here that if you draw a line through there, it doesn't match up with where you would think the bulk sediment is. So this suggests that some of that younger material is degrading from the continents, but the older terrigenous material that has a higher activation energy to be degraded is actually what's mixing in and being preserved with the marine sediment. Since then, we've analyzed some of our sediments. We haven't deconvolved these into activation energy yet, but you can see that we're kind of in this sort of triangle area. And what this allows us to do, this technique allows us to do, and this is our selling point to NSF, is this allows us to sort of rewrite the carbon budget on continental shelves in these types of systems that are reworking the terrestrial organic matter. And based on our preliminary results, we're looking at changing that somewhere between 40 and 100% in terms of how much terrigenous organic carbon it actually preserves compared to what we thought in the past. So at that, I thank you. I'll take any questions if anybody wants to. If not, we can go ahead.